Um, um, with radiation is bad. Radiation isn't good for me. You should sometimes we have high doses, we have low doses. The radiation you are being hit with right now in the highest concentration is probably going to be radio radiation or radio waves and then visible light. Violet radiation, what we are going to focus on is visible light radiation. There's a very specific light. Because these are the ones that convey the most information. This range of light, or this range of wavelengths. This range of wavelengths is unoccupied molecular results in an action potential. Interpret as visible light. Now there are radiation near the red wavelengths, and red wavelengths are going to be low and when we think this ultraviolet radiation is typically considered perceived. Most of us are going to associate radiation. It's not for us. It'll cause two adjacent thymine molecules to be merged together. That can cause point mutations within our genome as we form thymine dimers. Now, in the most anterior portion of our eye, and directly inferior to the cornea, we have this watery liquid called the aqueous humor. Our aqueous And as this aqueous humor is body, where we have a venous sinus that reabsorbs aqueous humor. Even though the lens functions to have light travel through it, and for us, when we think of lenses, we think of things made of glass or made both prone to injury. When people accumulate damage to the lens, oftentimes this damage is going to be cumulative over their lifespan. Our lens is ciliary body. And as the muscle of the ciliary body contracts and relaxes, it's going to pull on the segments as the lens change shape. Seeing those photons of light. Just posterior to the lens, we have the next chamber, the posterior segment. And this posterior segment is filled, it's very jelly-like, called vitreous humor is literally the liquid of life. And as we look at this vitreous humor, that's slightly different optical properties compared to the aqueous humor, and this aids us with focusing light in the back of our eye. We have the retina. And there's some key components of the retina that we need to talk about. And optic disc, those are the two most notable structures on the back of the retina. The retina on the back of the eye is going to be filled with photoreceptors. And within the retina, there's a region that's very, very densely populated with photoreceptors called the macula lutea. Overwhelmingly of color photoreceptors, or cones. We'll talk more about the cones and the rods in a little bit. When we talk about those cones at the macula lutea, I want you to think cones for color, C and C, cones for color. And we have lots and lots of cones in the macula lutea. In the very center of the macula lutea, we have the density of only cones. And this region with an incredibly high density of only cones is called the fovea centralis. And this makes up a focal point. When you are looking directly at the macula lutea, and we have it's kind of, a give and take as a compromise. Then your peripheral vision will be incredibly high density of photoreceptors is we need a lot of processing power. If we had fovea centralis in the macula lutea, to process the image. So instead, we compromise. We have muscles so that we can move our eyes around and focus on something specifically, and then not have to waste all that brain power processing peripheral vision. Disc or blind spot. Let me go back a slide for us here. If we look right here on our retina, there's an area where we have lots of blood vessels over the surface of the retina. This area with lots of blood vessels over the surface of the retina is called the optic disc. And because those blood vessels are covering up the surface of the retina, they block photons of light. So when we think of the optic disc, we can't perceive any vision there. The photons of light can't get to any photoreceptors. Our optic disc is often known blind spot test we'll project it on the screen. If you printed off your PowerPoint slide, you would need to hold it about at arm's length in front of you and focus on the X and then wiggle it around a little bit and the red dot will disappear. It can happen, most people can have that happen within a matter of seconds. It's also in your lab manual as well. I would be remiss if I didn't mention that several forms of martial arts teach you to punch within the blind spot of the opponent. The human blind spot is about right here where my fingers are. And I remember very specifically when my younger brother learned how to punch in the blind spot. I literally didn't see it coming. So let's talk about our pupil and our iris. Our iris controls how much light gets into our eyeball. It can contract, relax. I remember as a younger student, I was confused about how you can open the iris by contracting a muscle and how you can cause the iris to get smaller, how you can close the iris by contracting a muscle. And the reason we can open and close the iris by contracting a muscle is because we have two separate muscle groups in the iris. When you contract one versus the other, you can open versus close the iris. So as we look at our iris, we have the pupillary dilator. Our pupillary dilator is going to have muscle fibers that radiate outward from the central point, from the pupil. Our pupillary constrictor is going to be a sphincter muscle that encircles the pupil. When we contract the pupillary dilator, it's radiating muscle fibers contract. And then when we contract the pupillary constrictor, we are going to contract that sphincter, that circular muscle, and we'll narrow the pupil, we'll make the pupil opening smaller. This is important because we don't want to overwhelm the photoreceptors. We can overpower photoreceptors and we kind of bleach everything out. You probably experienced that sometime at night when you're driving your car and then somebody hits you with their eye beam. Because you're that and then an incredibly bright light is shown into your pupils. On the flip side, in low light situations, we open our pupils wide open, and we open them wide open because we don't have a lot of low light receptors, and we're trying to get as much light as possible into our iris. So light intensity is very important for regulating the relaxing or the opening and the closing of our pupils. We're also going to have a little bit of regulation based on what we're focusing on. If we are focusing on very close objects, those very close objects cause us to shift our pupils just a little tiny bit. And if we're focusing on objects that are really far away, we tend to open up our pupils just a little bit more. It's analogous to the diaphragm microscope that we've been using. If you're looking at an image and it's really funny, you can look at the diaphragm on the microscope, and two things will happen. One, the image gets darker, less light gets into it. If it's darker, the resolution is on. And a lot of it has to do with the fact of photons of light in straight lines radiating out. Looking at something that's really far away, typically 100 meters away or farther, all those photons of light that you're looking at are traveling fairly parallel to each other. But when you're looking at something that's really close to you, like from your finger in front of your face, photons of light are hitting straight on, and other photons of light are traveling oblique to your face. And we close that pupil just a little tiny bit so that those sideways traveling photons make it into our eye. We have better image resolution. How many of you have gone to the eye doctor and had pupils dilated with eye drops. Then you walk out of the eye doctor and everything's all fuzzy. The reason everything's all fuzzy is because all of those oblique photons of light, your brain is having trouble processing your vision. Another key barrier we have with vision is the refraction of light. Next. When we are very concerned about the direction that light is traveling. The catch is when photon of light is at an angle other than a 90 degree or other than a straight on angle. And in those situations, the photon of light will be bent. You've seen this happen before, whether it's like fish in a stream, fish in a fish tank, causing glass stream. Photons of light can be bent depending on the angle of traveling and the refractive properties of an object. Some photons of light can travel really quickly. Some photons of light are slowed down. Depending on what the photon of light is traveling through, its speed will change and the amount of photon of light will change. The hard vacuum of space. And because we have air, we have water, cold air with high density, we can have low density hot air, or we can have humidity in the air. All of these different things influence the speed that photons travel and can influence the way that you perceive an image. The light is being bent. So in our eyes, these optical components of our eyes are part of this light that's passing through the cornea. So when the light hits the eye, first hits the cornea, some light is going to be bent. It's coming straight on. So this light is right in the center. We'll always proceed directly straight. But some of the light doesn't hit the cornea in the center. Some of the light hits the cornea angles, and that light needs to be bent or refracted through the pupil to the macula lutea, focus and trials on our retina. As we look at the aqueous humor and the vitreous humor, the lens, and all these optical components of the eyes, all have slightly different refractive indexes. For our frame of reference, our definition at sea level, at standard temperature and pressure, is defined as having a refractive index. We're a physics major. So as we look at the lens and the vitreous body and the vitreous humor, these different substances, these substances will have light traveling slower through them compared to air. So if we look at this vitreous body, because we have a number greater than 1, light travels slower through it than it does air. If we look at our lens will change shape. These segments of the lens will relax. And as these suspensor liquids relax, the lens becomes rounder, and a rounder lens is capable of taking oblique light that's traveling from an oblique angle and re-angling light to be focused on focus centralis. We relax the ciliary muscles. We round our lens because the light is traveling at a wide variety of angles, we need a wider curvature to our lens to accommodate that variety of photon travel. And sometimes this happens automatically. Think of the last time you're reading a really good book at night. And you're reading, you just put down a page turner. That happens to the book here. And usually the one reading that might take and they start to relax. As your eyes relax, the lens is going to become out of focus, and then the eyes. So during this eye relaxation, you're going to be following objects farther than 6 meters. For these objects that are coming from really far away, in a straight line, there's very little deviation. There's still going to be a little deviation at that 6 meter mark, at 100 meter mark, it's straight. Now, for that, those rays of light that are coming from objects that are closer, those are going to be to accommodate for the traveling in direction. To accommodate. First, we're going to take our eyes and change the direction they're pointing. We'll try to center our eyeballs on the pathway of light travel, so that there's this little lens, so on and so forth. So we can reopen our eyes to point at what we're trying to look at. Constricting the pupil, eyeballs, or simply only going to let the straight shooting photons, the photons that are traveling in a straight line, will get into our pupil and back into our lens. When our pupil is wide open, we have more light photons traveling in multiple directions, and it's harder to focus on our image. And then finally, our lens. We are going to change the shape of our lens, change the curvature of our lens, that far away. So as we're looking at our lens, when we're trying to look at something that's close, we want the lens to be thicker. We want more curvature to our lens. As we're trying to look at something that's far away, we constrict the ciliary muscles, pull on the suspensory ligaments, and we can change the focus of the lens. Sensory transduction. This is going to be detail. There are two kinds of photoreceptors in our eyes. We have the rods and we have the cones. They're called rods and cones because of the shape of the outer segment. The outer segment of the rod, well, looks like a rod. The outer segment of a cone looks like a cone. I want to associate rods with low light. We use cones for color vision. Both of these photoreceptors are going to be embedded within the retina. And within our retina, we have a couple layers to it. We have our first layer directly against the vitreous humor, known as the, we have our posterior layer farthest away from the vitreous humor, known as the pigment epithelium. The purpose of our pigment epithelium is to keep light from hitting the photoreceptors from the backside. We only want our photoreceptors to be activated with light that travels through the portion of our eye. We don't want a backdoor photon causing an action potential. We're also going to have the rods and cones, those light absorbing cells. And as we look at those light absorbing cells, there's two different pigments in them, depending on whether it's a rod or a cone. Within rods, we are going to have rhodopsin as our visual pigment. This is the molecule that is highly sensitive to light. It's prone to changing shape very easily with low concentrations. Within our cones, for our color vision, we're going to have three different flavors of cones. These three different kinds of cones within human beings are going to focus on different wavelengths of light, and by mixing and matching these three different kinds of cones, we can perceive the rainbow of colors. This is the key difference between a rod and a cone, in addition to the shape of its outer segment, and that's going to be rods have rhodopsin, RR, rods for rhodopsin. For our cones, for our color vision, instead of rhodopsin, we have a pigment molecule known as photopsin, sometimes referred also to as iodopsin or iodopsin. Proteins between our three different kinds of cones are going to have sequences are going to change which specific wavelength of light is being absorbed, whether it's a wavelength of light closer to the 400 nanometer end of the spectrum or the 700 nanometer end of the visible spectrum. If we were to look at the histology of our retina, we have the anterior portion on the lower part of the screen, the posterior portion is on the upper part of the screen. We have the sclera as the most posterior part of the eye, the white part that wraps around the outside of the eye. Next, we'll have the body, which is a very pigmented dark layer filled with many blood vessels. And then for the retina in particular, we have the pigment epithelium. It's worth mentioning that in human beings, our pigment epithelium is kind of boring. It's not very exciting. But some animals that are optimized for low light vision are going to have reflective pigments within their pigment epithelium. And those are animals that you can see glowing eyes. So I'm thinking of cats in the ditch, because I grew up in the countryside. And we would be driving my car at night, and I'd see these two glowing eyes looking out at me from the ditch. And the reason those organisms have the glowing eyes is because their pigment epithelium contain reflective pigments. We as human beings don't have reflective pigments in our pigment epitheliums. Our eyes don't glow at night, thankfully. As we keep moving anteriorly, we're going to have the photoreceptors. So we'll have the rods and the cones. And then we're going to have some cell types that help process our vision from the rod and the cone. We're first going to have a layer of bipolar cells, and then we'll terminate with a layer of ganglion cells. One thing that's worth emphasizing about human eyes compared to other animals within the animal kingdom is that human eyes don't have photoreceptors directly on the surface of the retina. Other animals within the animal kingdom are going to have the rods and cones directly on the surface, directly on the anterior surface of the retina. Those organisms have better vision than human beings. Multiple layers of cells blocking vision, blocking photons from getting to our photoreceptors. So compared to other organisms, our vision is comparatively lacking. Let's talk about rhodopsin. Rhodopsin is the pigment within our rods to focus in low light environments for monochromatic vision. And in a dark environment, or when rhodopsin is not being exposed to light, it is going to be in the transretinal state. We find that rhodopsin, retinol, will work together. When we expose this molecule, expose the rods to light, we find that our retinol molecule will straighten out. So it goes from the cis state to the trans state. Cis and trans are organic chemistry terminology that refer to the carbon state. Those carbons are going to be on the same side of the double bond within the carbon, and when they're in the trans state, those carbons, or the backbone, will be on opposite sides of the double bond within the carbon. For our purposes, though, when we look at cis versus trans, we're looking at a bent molecule versus a straight molecule. When we straighten out that molecule, when we expose that molecule to a photon of light, it straightens out and initiates the cascade that starts our action potential. But it takes a while for that molecule to rebend itself. When we hit it with the photon of light, we energize it. It straightens out, it goes to the higher energy state. It takes a while. This period of time that it takes for it to relax back down to its lower energy state is a period of time when we have trouble seeing. So that's you have the bright light shown in your face. Typically a couple seconds. So you're young, so it's probably going to be more in the seconds as opposed to the minute end of the spectrum. But for a couple seconds, you probably don't see anything. And that's because all of your photoreceptors within your rods were in the straightened state. They were in this trans state. And it takes a while, typically, for them to reorient, rebend themselves, and reposition themselves so that they can receive another photon of light. When we think of the cones within our vision functions, we have photopsin. That's going to focus and function very much the same way. It's going to bend in response to the photon of light. Now, as this rhodopsin, sometimes referred to as retinol, goes from bent to unbent, it's going to change its shape. By changing its shape, it's going to change its chemical activity. And by changing the chemical activity, we influence the secondary cyclic AMP, the calcium ions, and can initiate that action potential. It all comes back to structure and function. I think we talked about on the very first day of class. Structure of something determines its function. When retinol is straightened out, secondary messaging cascade and starts the action potential. When it's bent, it does not start that secondary messenger cascade, and we don't perceive a photon of light. Now, in the dark, our rod cells are going to still be seen a neurotransmitter called glutamate. Sometimes this glutamate also functions as an amino acid. In this context, it's a neurotransmitter. When our rod cells absorb light, they're released. There are two other layers of cells we talked about. We have the bipolar cells, and then we have those ganglion cells. And they serve a very important purpose. The rod leads up to a bipolar cell. When our rod stops releasing glutamate, our bipolar cell becomes activated. And then our bipolar cell will start to release neurotransmitters that go to the ganglion cell. There are a couple variations of ability to see in low light versus high light situations. Some of our bipolar cells are going to be inhibited by glutamate. Some are going to be excited. When we focus on rods, I want us to primarily focus on potential for our brain. They're not going to focus on ganglion cells. vision is because there's a trade-off with our rods and cones. Distinguishing details. Point in our eye has a very high concentration of cones and almost no rods. And we're also trying to conserve processing power as well. Within our brain, we don't want to spend a lot of time processing an image that we don't care about. What do we care about from an image perspective? We typically care about the image that we're looking directly at. So whatever we're taking on our focus lutea, that's what we're arbitrarily caring about at that moment in time. So as we look at high-resolution vision of the lutea and fovea centralis, and that low-resolution peripheral vision, we have different circuitry associated with them. So let's talk about low-resolution vision. Low-resolution vision is typically going to be associated with peripheral vision. And in these situations, we have a lot of neural convergence, which is kind of a nice way of saying that we have lots and lots of photoreceptors for an individual ganglion cell. We'll have many rods, up to 600 rods, converging onto one bipolar cell. And then those bipolar cells can all converge onto a single ganglion. This results in well over 1,000 rods being associated with one ganglion cell. In other words, we have over 1,000 different photoreceptors all producing the same one action potential. We don't spend a lot of brain power differentiating signals from different cells when we look at it with our rods. You've probably experienced this before. When you think of this dim light or low resolution vision or peripheral vision, think of those times when you've been looking at stars at night. And if you try to look directly at the star, a lot of times it disappears. But then if you look off to the side, it will reappear. And the reason those stars or those very dim objects tend to reappear when you're not looking at them is because in our peripheral vision, or around the edge of our retina, we have lots and lots of rods with low light sensitivity. You probably all, like another example of this would be maybe you're in your bedroom at night and you see something that's kind of freaking you out in your peripheral vision and you turn to look at it and it completely disappears. You can't see anything at all. And you look away and you can see it again. The reason for that difference has to do with concentration. Light neural convergence of precision vision. We typically have 1 square millimeter of our retina being served by one individual ganglion cell within our peripheral vision. The fovea centralis. Our fovea centralis has only cones, 0 rods. And because we have only cones in our fovea centralis, we are only able to perceive images that have lots of light intensity. We can only look at things that are bright with our fovea centralis. And if we look at the fovea centralis, we don't have any neural conversion. So if we back up a slide here, we can have well over 1,000 rods per ganglion cell in our peripheral vision. In the fovea centralis, it's a 1-to-1 ratio. 1 ganglion cell per cone. 1 active per to process a much higher resolution picture within the fovea centralis. The downside, having only cones in our fovea centralis, is that we as human beings have no low light receptors, no rods in our focal point of our retina. Let's talk color vision. When we think of color vision, we have 3 different kinds of cones in our eyes. eyes. These three different kinds of cones are going to perceive three different wavelengths of light. We have the short, the medium, and long wavelengths that correspond cones that correspond to the length of the wavelength of light perceives. The short cones are typically going to be referred to as the blue cone. We have the medium wavelength cones that perceive green, and then the long wavelength cones that perceive red. It just so happens that this blue, green, and red is typically the same three colors that you see within a pixel on a computer screen. Now, most of your computer screens, most of your cell phones have pixels that are too small to see with your naked eye. But back in my day, we had low resolution everything. So I can remember very specifically, you could put your head up in front of the giant TV screen and see individual pixels. You can with your cell phone. You just need to have a drop of water on your cell phone. And that drop of water can magnify a pixel, and you can see the red, green, and blue that make up that individual pixel. And our eyes can take red, green, and blue and turn those different signals into all colors of light. It's also worth emphasizing that between our medium and long cones, generally speaking, right about here, we have peak sensitivity. That peak sensitivity for color corresponds to safety green. That is the color, the wavelength of light that will cause the most photoreceptors, the most cones to be activated in our eyes. And that's why safety green tends to jump out at us so much, because we're activating so many of our cones. So as you mix and match the intensity of your three different kinds of cones, you can come up with different color perceptions. Was it 2 years ago? One of the big debates on social media was, is it blue or is it gold? Referring to a stupid dress photo. You probably remember that. And then if you read some of the follow-up articles on that, they talked about how some people have a fourth cone or fourth kind of color photoreceptor in their eye. There's no solid for that. I just wanted to go on record and say that. Let's talk about depth perception. Because we have two eyeballs on the same side of our head, we can have stereoscopic vision. We can have depth perception. You may have noticed that if you put your finger in front of your face and open and close different eyes, they move relative to the background. This process of having the finger move back and forth can be referred to as parallax. And your brain will use parallax from an object that's close to you to determine depth perception. If there's more parallax, it's going to be perceived as closer to you. If there's less parallax, it's going to be perceived as farther away from you. Organisms that have the eyes on the left and right side of their head, think of like mice or horse. Usually herbivores will have this. Those organisms don't have stereoscopic vision. They have 360-degree vision. They can see stuff coming from any direction. We as human beings tend to be a little bit more predatory in our dietary habits, and we typically need to focus more on seeing a specific object and honing in on it. When we look at an object that's far away, have less parallax, and our eyeballs don't need to diverge as much. When that object is closer to us, there's more parallax, and our eyeballs diverge more. And then we have our last review question of the semester. Why do cones exhibit less neuronal convergence than rods? Our options are: rods cannot distinguish colors from each other, rods do not function in bright light, no images are perceived with an optic disc, cones do not detect black and white, or photoptic vision has higher resolution than scotoptic vision. Neural convergence, multiple photoreceptors on cell. Make sure to get your answers in. Less than 5 seconds left. The correct answer was that photonic vision has solutions. Optic vision are cones function for photonic vision, or high-resolution vision. Everyone's present today.